Hello, 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 Janvi. Uh, Hello everyone. I am Nilanjana. I am a TA under Professor Himanshu Kumar. So, uh, he is teaching the topic of host interaction, host and microbe interaction, and basically he's teaching all the different types of uh, immunological aspects. So, this is the fourth week of NPTL, and this is supposed to be a doubt clearing session. So if anyone is having any doubt, they can post in the comment in YouTube as the link will be given. So sir is already teaching every topic of our immune system, but since no one is actually posting about any doubt, so I am basically taking few more, like some of the topics regarding immune system, and again I am teaching the same topics. So today we will be learning about uh, innate immunity. I hope everyone can see my slide. So the previous classes we have learned about innate immunity, adaptive immunity in brief. Now, like now we will be knowing about the different types of components of the innate immunity in broader sense. So here we can see a beautiful picture where this is a macrophage that is yellow in color, binds and is phagocytosing E. coli bacteria, red. So we all have learned what are the cellular component of innate immunity, like macrophage, dendritic cells. These are the neutrophils. These are the cells involved in innate immunity, and whenever there is a infection, these are the prime cells which come into the picture and help clearing the infection. So this is a macrophage binding and phagocytizing the E. coli bacteria, which might have entered into our body. So what are the we know that innate immunity comprises of many different things. So what are the different types of uh, function or barriers to infection which comprises of the innate immune system? So first is the physical and chemical barriers to infection. Like we know we are having skin. So there are different types of layers in skin present. So the outermost layer, which is the epithelial layer of the skin, and also the glandular tissue or the mucosal tissue in our body helps us in clearing the pathogens whenever they are entering. So also some acidic pH and antimicrobial peptides and proteins which are secreted by our skin or other layers like which are in the outer layer, like it is the primary barrier to infection. Whenever there is a some pathogen is trying to enter, it has to go through the skin or the epithelial layers or the mucosal layers. So it has to go through that layers to enter into our cells. So this is the primary first barrier for infection. So if there is any break in the epithelial layers, here we can see a break in the epithelial layers. So here what happens, the pathogen can enter because there is a break, so it can enter into our system. But after entering also, the second type which is coming into the picture are the different types of cells of our innate immune system, like the macrophage, dendritic cell, then natural killer cell. So what happens? There are receptors present on these cells which are binding to these pathogens. So after these pathogens are binding to the receptors, there will be either phagocytosis and degradation, direct degradation of the pathogen, or it can activate cells which can trigger the secretion of cytokines and chemokines, which I have already taught, like small molecules which can directly kill or activate other types of cell types. And this is natural killer cell which can directly kill these pathogens. So these cytokines and chemokines, here we can see that can activate the T cells as well as the B cells, which will produce the adaptive immune response in our body, and all this will lead to pathogen elimination. So, whenever we are eating food, so food is going through our mouth. So, when food is going, there is a mixture of bacteria. You are you think like whatever food we are eating, obviously it cannot be free of bacteria. Outside, so millions and millions of bacteria or viruses or whatever pathogens are there. So what happens then? Then ideally every pathogen should enter into our body. But this is not the case here because there are very much layers present which is eliminating the pathogen. So this is an overall view of our innate immune system, which can function at various levels to stop the pathogen from entering into our body. So, before starting, obviously, it is known what are the differences between innate and adaptive system. Like for innate immunity, it takes only minutes or hours to respond to the pathogen, while in adaptive immunity, it takes days. In case of innate immunity, it is specific for different molecules or patterns associated with the pathogens or the dead or damaged cells, while adaptive immunity is highly specific. It can discriminate even between major differences in the molecular structure of microbial or non-microbial molecules. In diversity also, it is already germline encoded, means it is present from the birth. We cannot change that after our birth. It is already germline encoded. But in case of adaptive immunity, it is very diverse. Like it can generate a number of receptors by genetic recombination of the genes. So, by permutation and combination, it can generate a large number of receptors. It is not inborn. So, with different types of pathogens, it can act differently. The memory response is not there in case of our innate immune system, except in some invertebrate and human NK cells. And in case of adaptive immunity, it has persistent memory, like with faster response of greater magnitude on subsequent exposure. So whenever there is a subsequent exposure of the same pathogen, our adaptive immune response will respond much faster compared to the previous exposure. The non-self and the self and non-self discrimination is very good in case of innate immunity, but in adaptive also it is good in case of adaptive immunity, this non-self and self discrimination. But sometimes when our immune system fails to recognize what is the self molecules and what is the non-self molecule, then there is a chance of some disease which call as autoimmune disease, which where our cells attack our immune system attacks our own cell. And soluble components of blood in innate immunity include antimicrobial peptides, proteins, other materials like cytokines, while adaptive immunity include antibodies secreted by the B cells and cytokines. Major cell types in innate immunity include phagocytes, natural killer cell, leukocytes, epithelial cells, and endothelial cells, while in adaptive immunity it is antigen presenting cell, T cell, B cells, etc. So these are the major differences between innate and adaptive immunity. Hello, Janvi. If you are having,
Hello.
if you are having any doubt regarding to what Sir has taught, you can ask.
Okay. Ma'am, I'm really sorry for the inconvenience. Right now, I'm somewhere outside, but I
Okay,
just
okay,
got reminded of our lecture.
okay, okay, okay.
Uh,
No
no, ma'am.
worries.
Actually, right now you're teaching about innate and adaptive immunity, and I'm having my doubts clear regarding this. If you are about to proceed further, and if I will be having any doubt, I will be asking you.
Yeah, yeah,
Thank you.
sure. Or you can type also in the comment, like
Okay, ma'am.
message box.
Okay.
Huh.
Okay, ma'am. Thank you.
So, what are the anatomical barriers to infection? Like, as I already told, the skin is one of the example. Like, not all pathogens can invade our skin and enter. So, there are different types of uh, passages through which the pathogens can enter, but there are barriers to to these pathogens which protect us from these pathogens. So, one is skin. So skin secretes some antimicrobial peptides or fatty acids which are present in sebum. Okay, so what are the anatomical barriers to infection? Like skin, it secretes many antimicrobial peptides, or in sebum, there is presence of some fatty acid. Mouths and upper alimentary canal, like as I already told, whatever we are eating is not free from pathogens. So there are certain barriers in our mouth and the alimentary canal through which the food passes. There they also secrete like enzymes, antimicrobial peptides. Then they also sweep like surface by directional flow of fluid towards stomach. So all the microbes which are entering will be swept through some cilia which are present. Then there is a stomach which is having low pH, digestive enzymes, bile salts, antimicrobial peptides, fluid uh, also flow towards the intestine. Then small intestine is also having the same, which is fluid flowing to the large intestine. Then large intestine have normal intestinal flora, our gut microbiome, which competes with the invading microbes, and also the fluid or the feces which are coming from the mouth and the upper alimentary canal is now flowing from the mouth to the stomach to the small intestine, large intestine. Then it is expelled from the rectum. Then in airway and lungs, there are cilia which sweeps the mucus outward. The coughing, sneezing expel mucus. There is macrophages in alveoli of lungs which prevent this, which phagocytose these pathogens whichever are entering through our airway or the nostrils. So there is also urogenital tract, like there is flushing of these pathogens by urine, mucus, there is low pH, there are secretion of antimicrobial peptides, proteins. There is also salivary gland, lacrimal glands, mammary glands, which are also flushing this by secretion and mucus and also antimicrobial peptides or proteins. So every layer of our body or every part of our body has certain functions like which secretes these peptides or proteins or through mucus, it is expelling the pathogens. So you can understand like how much our body is like specialized to keep these pathogens away. So here these are the parts. These are the different anatomical barriers to infection. So the first step the pathogen has to do is to cross these anatomical barriers. Only after it crosses these anatomical barriers, it can enter into our body. The first step is just crossing these barriers. Otherwise, you can think like there are so many pathogens out there. So if there are no like anatomical barriers in our body, then how much prone we are to different kinds of infection. But in reality, this is not the case due to the presence of these barriers. So our epithelial barriers prevent the pathogen entry into our body's interior. As I already told, the first and the most important, the skin, which our outermost physical barrier, it consists of two distinct layers. One is outer layer, epidermis, and the thicker inner layer, which is the dermis. So the epidermis contains different types of tightly packed epithelial cells, and its outer layer consists of mostly dead cells filled with a waterproofing protein we all know, which is termed as keratin. And the dermis inside is composed of connective tissue, blood vessels, hair follicles, sebaceous gland, sweat gland, and other leukocytes such as dendritic cell, macrophages, and mast cells. So, all these cells or all these glands will secrete some proteins and micro proteins and certain other antimicrobial substances which will prevent the entry of these pathogens. Then other epithelial surfaces of our respiratory tract or the gastrointestinal tract or the urogenital tract and the ducts of salivary gland, lacrimal gland like tears which we are secreting from our eyes or the mammary glands, they are also lined by these epithelial cells which are also stitched together by tight junctions that prevent pathogens from squeezing between them to enter the body. So whenever there is a cut, whenever there is any like small breach in this epithelial layer, there is a chance that pathogen can enter into our body. So also, the mucus, the viscous fluid which is secreted by our mucosal epithelial layers, also entrap the foreign organisms. Mucins, which are glycoproteins, are found in this mucus, and they can prevent the pathogen adherence to epithelial cells. There, in the lower respiratory tract, as I told, there are certain hair-like structures which are known as cilia. They are the protrusion of the cell membrane and cover the epithelial cells. So they move synchronously so that they will trap these microorganisms which are trying to enter through our respiratory tract, and will send them away. They remove them. Then coughing. Coughing is actually a good response for us. Coughing is a mechanical response that helps us to get rid of excess mucus with trapped microorganisms that occurs in many respiratory infections. So coughing is not always bad. There is some reason why we cough, and it helps in expelling this trapped microorganism from our uh, respiratory tract. And also urine also helps in removing many bacteria from our urinary tract. So there are these normal processes of our day-to-day life actually is helping us to remove so many pathogens which are entering into our body. So uh, there are certain organisms which are evolving, like we are evolving, like there are organisms or pathogens which are also evolving, and they want to evade our defenses of these anatomical barriers. For example, two examples I have given. One is influenza virus. So influenza virus has a surface molecule that helps it to attach to the cells in the mucous membrane of the respiratory tract. So we all know that influenza enter like is a respiratory virus. So it wants to attach to the respiratory tract in the mucous membrane. It will attach. There are some receptors. So, but as I already told, there are cilia present in our respiratory tract, which will prevent this virus and will help to get rid of this virus by sweeping them away. Another is one bacteria, Neisseria gonorrhea, that causes a disease, gonorrhea disease. It binds to the epithelial cells in the mucous membrane of our urogenital tract. Uh, but there are certain hair-like protrusions on the bacteria called fimbri or pili, which adhere in our urogenital tract, and there are certain glycoproteins or glycolipids present to which it adheres. Uh, so what they do is like they have evolved this fimbri or pili that to which they attach so that they can bypass our immune system and they can enter into our like urogenital tract. So there are like we are evolving every day. These pathogens also are very clever. They also want to evolve, and they also form different kinds of. They modify their structure so that they can infect our body. So here we can see electron micrograph of E. coli bacteria, which is attaching to the surface of urinary tract. Like these yellow are the E. coli, which are attaching to our urinary tract. So, um, so what our function of the epithelial layers are? It insulates the interior of the body from outside pathogens, the skin and epithelial layers of the mucosal tract and secretory gland. It constitutes an anatomical physical barrier that is highly effective in preventing pathogens from entering the rest of the body. So constantly there is a like removal of these pathogens from our body, while pathogens also try to evade this system by constantly evolving their structures. So as I already told, the skin, the respiratory tract, the urogenital tract, everything secretes some antimicrobial proteins and peptides. So, what are the different types of antimicrobial proteins which are present? If anyone is having any question, they can ask me in the chat. So, the different types of antimicrobial peptides and proteins are lysozyme. So, lysozyme, as we all know, are present in tears, saliva, respiratory tract. They cleave the glycosidic bonds of peptidoglycan in cell walls of bacteria, leading to lysis. So there are lactoferrins, which also are secreted through the milk, intestinal mucus, nasal, respiratory, and urogenital tract. They bind to and sequester iron, and iron is very much needed for the growth of the bacteria as well as fungi. So they, for the pathogens, it is very much needed. So they uh, binds to this iron and sequester this iron so that they cannot grow, the bacteria cannot grow, and they disrupt microbial membrane. They also limit infectivity of some viruses. Then there are secretory leukocyte protease inhibitor, which are present in skin, mucosal, granular secretion like intestine, respiratory, and urogenital tracts and milk. They blocks epithelial infection by bacteria, fungi, viruses, and they are mainly antimicrobial in nature. There are proteins like S100 proteins, sodiacin, and calprotectin. They are also present in tear, saliva, intestine, respiratory, urogenital tract. They disrupt the membrane, they kill the cells. They also bind and sequester divalent cations like manganese and zinc. They limit the growth of bacteria as well as fungi. There are proteins like surfactant proteins. They are also secreted by respiratory tract, mucosal epithelia, and they block the bacterial surface components and promote phagocytosis. Other proteins are reg3 proteins, intestinal, which are present in intestinal epithelia. They bind to the cell wall and carbohydrates and prevent bacteria binding to the epithelial cells. They also produce some pores in the membrane that can kill the cells. So these are the antimicrobial proteins. And what are the peptides? The peptides are, I hope you all know the difference between proteins and the peptides. Like proteins will be smaller, but the proteins will be larger. Like when peptides join together, it will form large proteins, but peptides are smaller in size. So different kinds of peptides like defensins alpha and beta, which are present in the mouth, intestine, respiratory tract, urogenital tract. They disrupt the membrane of the this parasite, fungi, bacteria, viruses, and they kill cells and disable viruses. There are also cathelicidin that is present in the mucosal epithelia, example respiratory tract and urogenital tract. They disrupt the membrane of bacteria and additional toxic effects intracellularly and kill cells. There are histatins which are present in the saliva. They bind to fungal cell walls and enter the cytoplasm where they have several harmful effects. There are also peptides which are present in skin known from the sweat gland, that is dermicidin. They are also antifungal and antibacterial, and they also produce some channels in the membranes that disrupt these ion gradients. So these are the different types of antimicrobial proteins and peptides which actually which are secreted from the different anatomical barriers of our skin and kills the pathogens which are trying to enter into our body. So one experiment was done. Here we can see like sodiacin, as I already told, is an antimicrobial proteins, but these are very much specific to some organism. For example, one experiment was done where they saw that sodiacin, that antimicrobial protein, prevents the colonization of the skin by E. coli. And while it is not effective against Staphylococcus aureus. So, as I already told, that sodiacin is secreted by this tear, saliva, skin, urogenital tract, intestine, nasal, respiratory tract. So this sodiacin, what they did was they have inoculated uh, they have inoculated they have taken the healthy human fingertips, and they, in one hand, they inoculated the fingertips with Staphylococcus aureus, and in another hand they inoculated with E. coli. And after 30 minutes, they were the fingertips were made to press on a nutrient agar plate. So, uh, in this agar plate, the agar is kind of a food to the bacteria. So, what they did, they just put some Staphylococcus aureus in one hand, another in one hand, E. coli they have put. And after 30 minutes of incubation, they have just told them to press their fingertips on the nutrient agar plate. Then they saw that in case of the fingers in which they have inoculated with E. coli, there was no colony in the plate, almost nil the colony number. But in case of Staphylococcus aureus, like it was, there were so many colonies. So here also we can see they have inoculated this with Staphylococcus aureus and E. coli. They inoculated for 30 minutes, then they press the fingers, both the hands, on the nutrient agar plate. Then they incubated it overnight, maybe, and then they saw how many colonies. So here we can see in E. coli there is no colony, only two we can see. But in Staphylococcus aureus, you can see the fingertips, like the shape of the fingers. There are so many colonies here. So what it proved, that sodiacin is secreted by our hand. So whenever there is a E. coli in our hand, this skin will secrete sodiacin. So this sodiacin will kill this E. coli bacteria. But whenever there is Staphylococcus aureus, it won't kill that bacteria. So sodiacin, they proved that sodiacin is specific for E. coli. So all these antimicrobial peptides are can be specific to different kinds of organisms, and not all can act against all the microorganism, but some can act against all the microorganism. It depends on the uh, type of the protein as well as the microorganism. So antimicrobial peptides, as I told, like, they differ from the antimicrobial proteins because they are smaller in length. They are generally less than 100 amino acids long, and they are generally rich in cysteine residues. They are cationic, they are amphipathic, that is containing both hydrophilic as well as hydrophobic residues. Because of their positive and amphipathic nature, they can interact with acidic phospholipids in lipid bilayers, forming pores and disrupt the membranes of the bacteria, fungi, parasites, as well as viruses. However, they can also enter into the microbes, where they have toxic effects, like they can inhibit the synthesis of DNA, RNA, protein, and activate antimicrobial enzyme, resulting in death. So the different types of antimicrobial peptides, as I already told, are beta defensin, alpha defensin, cathelicidin, histatins, dermicidin. So only the difference between peptides, antimicrobial peptides, and proteins are they are just smaller in size, in the amino acid length, compared to the proteins, antimicrobial proteins. So after this, like anatomical barriers, suppose are breached, like there is cut or there is any kind of breach by the anatomical barriers, by the pathogens, as I told, that they also evolve to enter into our cell. They also evolve their different uh, properties, like they change their ligands, they change their affinity. So whenever there is a breach in this epithelial layers or the other layers and the other barriers. So the pathogen will enter into the cell. So what happened after that? After that, it should go and interact with the receptors. So there are different types of cellular innate response receptors which are present in our body, and mostly these cellular receptors are known as pattern recognition receptors. So they are having some essential role in detecting the presence of pathogen and activating the innate immune responses that combat the infection. So these PRRs, or the pattern recognition receptors, they bind to certain molecules known as pathogen-associated molecular patterns that trigger cellular responses. So whatever pathogen-associated molecular patterns means what? Pathogens are having different kinds of molecules on their surfaces. So they are acting as a pattern to these receptors. Like whenever these patterns are found inside our body, these receptors will get activated and these receptors will bind to these patterns or the molecular patterns which are present in the pathogens, and they will trigger the immune response, downstream immune response. So all these PRRs are present, some are present on the plasma membrane where they can bind to the extracellular pathogens which are coming. And others are present inside our cells in the endosome or the lysosomes where they bind to the PAMs released by endocytosed pathogens, or in the cytosol where they respond to PAMs such as cytoplasmic bacteria and nucleic acids from replicating viruses. So there are endosomal PRRs, there are PRRs present on the plasma membrane. So in the plasma membrane, they can directly bind to the extracellular pathogen whenever there is a like there's an entry of any pathogen, they can directly sense that pathogen. But inside the endosome, they will bind to the molecular patterns which are released by the endocytosed pathogen, or in the cytosol where they respond to the uh, PAMs such as the bacteria nucleic acid from viruses. There, apart from these pathogen-associated molecular patterns or PAMs, there are DAMs or damage-associated molecular patterns. So the damage-associated molecular patterns are released by cell or tissue whenever there is any kind of tissue death or tissue damage or cell damage. So these are also recognized by our cell surface as well as endosomal PRRs. So there are two different types of ligands, PAMs and DAMs, pathogen-associated molecular patterns and damage-associated molecular patterns. So all these are recognized by our pattern recognition receptors. So first we will know one by one what are the different types of innate response receptors. First is the toll-like receptor, or the TLRs, which are found to initiate responses to many types of molecules from extracellular pathogens. So in 1980s, researchers in Germany discovered that the Drosophila fruit fly embryos, I hope you all know, Drosophila are the fruit flies, which we can everywhere, whenever like in the environment we can see Drosophila. Those Drosophila fruit fly embryos could not establish a proper dorsal ventral, means back to front axis, if a particular gene or encoding the toll membrane protein is mutated. They found that if this protein is mutated, there is no proper axis which is present in that Drosophila. No dorsal ventral axis is formed. So they term this as weird. It this toll means from a German slang it is coming meaning as weird, referring to the mutant fly's bizarrely scrambled anatomy. So this toll-like receptors, which is meaning weird because it when they mutated this protein, they saw a very weird kind of anatomy in this Drosophila. So after that, they found this that first they found that this toll membrane is responsible for this dorsal ventral axis. But however, in 1996, Jules Hoffman and Bruno Lemaitre discovered that whenever there is a like mutation in this toll gene. So these flies are very much susceptible to lethal infection with Aspergillus fumigatus, which is a fungus to which wild type flies were immune. So in wild type flies, these flies were immune to this kind of fungus, but whenever there is a mutation in the toll, they saw that these mutant flies were very much susceptible to this infection by this Aspergillus fumigatus. So here we can see that this is a fly of the Drosophila fly, and this is a mutant fly where there is a mutation in the signaling pathway downstream of the toll pathway, and there is this green or the yellow is the fungus Aspergillus fumigatus. So this severe infection of the fungus Aspergillus fumigatus results from a mutation in the signaling pathway downstream to the toll pathway of Drosophila that normally activates production of the antimicrobial peptide drosomycin. So they found that there is a peptide, antimicrobial peptide drosomycin, which is produced in the wild type of the Drosophila, which is not being produced in the mutant type. So the mutant type is more susceptible to the fungal infection compared to the wild type. So for this also, Jules Hoffman got the Nobel Prize also for discovering the this uh, function and the gene which is responsible. Actually, in Drosophila and human, many genes are identical. So many genes were discovered in Drosophila because in human it is difficult to previously, not now. It was difficult to do experiments in with live humans or that time cell lines were also not that much established. So people used to do experiment in Drosophila as it can be a human like a human model. They found out many things which are common to human as well. So they found like a similar kind of uh, gene is present in human which is having the same sequence, almost same domain kind of sequence, and they termed it as toll-like receptors or toll TLR in humans. So TLRs are membrane proteins that share a common structural element in their extracellular region called leucine-rich repeats. So these TLRs are having are rich in leucines, and they have these leucine rich repeats in the extracellular region. So multiple LRR make up the horseshoe shaped extracellular ligand binding domain of the TLR polypeptide chain. So multiple LRRs make up the horseshoe shaped this they will make up a horseshoe shaped ligand binding domain, and there is a extracellular domain which is coming out of the cell and the intracellular domain, and some portion will be embedded in the plasma membrane. So whenever there is a binding of these PAMs or DAMs via the extracellular LRR domain, they will dimerize either as a homodimer like TLR2 TLR3 will join together. Here we can see 2 TLR3 are joining together, and this is the binding of the ligand, which is a double stranded RNA, or it can bind to a different TLR like TLR2 and 1. Here we can see TLR2 and TLR1 binding, and there is like lipopeptide present. So uh, there is a either formation of homodimer or heterodimer by these TLRs, and you can see there is a TIR domain present in the cytoplasmic region which can bind to other TIR domain of the different types of adapters which are present for the downstream signaling. So, as I already told, there are different types of TLRs. Some are embedded in the plasma membrane, some are present on the endosome. So here in the plasma membrane, you can see there are different types of TLR, but all are forming either heterodimer or homodimer. So this is 2 TLR4 present, which can recognize gram-negative bacteria. This is TLR1 and TLR2, which are making a heterodimer. It can recognize bacteria or parasite. This TLR2 and 6 heterodimer, which can recognize gram-positive bacteria and fungi. This TLR5, which is forming a homodimer, it recognizes bacterial flagellum. Then this TLR11, which is present, 2 TLR11, which is forming homodimer, and it can recognize toxoplasma, uropathogenic bacteria. Then TLR12 can recognize toxoplasma profiling. And apart from that, in endosomes also there are present uh, like uh, uh, TLR4, which can recognize viral proteins, TLR3, which can recognize viral double-stranded RNA, TLR7, homodimers, which can recognize viral single-stranded RNA, TLR8 can recognize viral single-stranded RNA, as of TLR9, which can recognize bacterial or viral DNA or plasmodium. So all the TLRs, either homodimers or heterodimers, are recognized as different kinds of uh, molecules, different kinds of PAMs from different kinds of pathogen. Not every TLR can detect every pathogen, and not uh, like every pathogen is detected by the same TLR. So there are diversification in the recognition of the pathogens by these different kinds of TLRs. So we all know that there is a difference in the cell wall component of gram negative as well as gram positive bacteria. So here we can see, like in the gram negative bacteria, the peptidoglycan is very much thin compared to the gram positive bacteria. Gram negative is E. coli and gram positive Staphylococcus aureus. So the peptidoglycan is much thicker in case of gram positive bacteria. So we we differentiate between these two types of bacteria via uh, gram staining. So due to the presence of this thick peptidoglycan, it can retain that stain, that gram positive bacteria, while in case of this gram-negative bacteria, this peptidoglycan is much thinner and it cannot retain that stain. So, according to the staining property, we divide this bacteria into gram-negative as well as gram-positive. So, here also you can see there is presence of this LPS, which is not present in this gram-positive bacteria, and this LPS is recognized by a different type of uh, as here you can see TLR4 can recognize this gram-negative bacteria because it can sense the LPS, while this TLR2 and 6 can recognize the gram-positive bacteria. So there are different properties based on which these TLRs recognize these different kinds of bacteria. Uh, then, so depending on the different types of bacteria or different types of viruses, different types of TLRs can recognize different types of components present on that particular uh, pathogen. So here is a list of all the toll-like receptors and their microbial ligands. Like TLR1 can recognize triacyl lipopeptides which are present in mycobacteria and gram-negative bacteria. TLR2 can recognize peptidoglycan, lipomannan, lipoprotein, GPI-linked protein, zymosan, phosphatidylserine. So peptidoglycan is present in gram-positive bacteria. GPI-linked proteins is present in trypanosomes. Lipomannan lipoproteins are present in mycobacteria, other bacteria. Zymosan is present in yeast and other fungi. Phosphatidylserine is present in cystosomes. TLR3 can recognize the double-stranded RNA from viruses. Double-stranded RNA viruses it can recognize. TLR4, as I already told, it can recognize LPS from gram-negative bacteria. F protein from respiratory syncytial virus, G glycoprotein from vesicular stomatitis virus, mannans from fungi. TLR5 can recognize flagellin from bacteria. TLR6 can recognize diacyl lipopeptides from mycobacteria and gram-positive bacteria, zymosan from yeast and other fungi. TLR7 can recognize single-stranded RNA from viruses. TLR8 can also recognize single-stranded RNA from viruses. TLR9 can recognize CpG islands or unmethylated dinucleotides present in bacterial DNA and dinucleotides present in some herpes viruses. Herpes virus components such as which are present in malaria parasite heme byproduct as well as some other components known as hemozyme. TLR10 can recognize, it is not known, currently unknown. TLR11 also profiling from Toxoplasma gondii. TLR12 also same. TLR13 is rRNA from gram-positive bacteria. So all these, you can see that in this table, everything is like a common molecule they are recognizing. Like if it is recognizing LPS, LPS is present in all the gram-negative bacteria. So TLR4 can recognize all the gram-negative bacteria. It is not like a specific protein which is present in a particular bacteria, particular virus. It is as a whole, as a general, a whole molecular level, it is recognizing some molecule. So it can, under gram-negative bacteria, it can recognize the whole lot of bacteria which are present by just seeing the or sensing the LPS. So how broadly it can recognize, you can understand these receptors. So this is the TLR signaling pathway, like the plasma membrane TLR signaling pathway through mediated adapter. So there are two kinds of adapters through which this signaling takes place. One is mediated, another is strip. So in the plasma membrane, there is a, you can see, heterodimer of TLR1 and TLR2 present. It binds to a lipopeptide, which is a pathogen associated molecular pattern from a bacteria. So it activates the downstream signaling So whenever there is a binding of this PAM, it induces the dimerization of the TLR. So as I already showed previously, there is a TIR domain. This TIR domain is present in the cytosol. So this TIR domain present in the TLR also interacts with the TIR binding adapter. This is a mediated adapter and it initiates signaling by recruiting different kinds of kinases. Like there is binding of whenever there is a sensing of this pathogen, this dimerization, then there is a binding of this adapter IDATA, which is connecting this TLR to different kinds of kinases like IRAC4, IRAC1. So whenever there is a binding, then there is phosphorylation in IRAC1. Here we can see. Then after that, some other proteins like TRAC6 is recruited, and there is TAC1 complex, like TAB1, TAB2. These are different kinds of protein complexes which are required for the downstream signaling process. So adapter is just helping in transmitting the signal, downstream signal. So if there is a mutation in this mediated, it cannot go and bind to this TLR, and there will be no downstream signaling present. So after the recruitment of these protein complexes, there is two pathway. Here we can see one is MAP kinase pathway, another is the the NF kappa B pathway, which is happening. So in this MAP kinase pathway, it activates different transcription factors, which is known as AP1, and here also there is activation of this I kappa B. Like there is two different subunits, I kappa A and I kappa B. It gets phosphodilated, then it this I kappa B is one subunit which inhibits this NF kappa B, and thus it uh, inhibits NF kappa B. So whenever there is a phosphorylation of this I kappa B, it will lead to degradation, and NF kappa B can travel to the nucleus, and it can induce the transcription of several types of ISG or interference stimulated genes, or even other genes which are required for the activation of the immune response. So by the any component in the signaling pathway, if it is mutated, then the downstream signaling will be stopped. So this signaling is through the mediated adapter. Apart from this, as I already told, another adapter is the TRIF adapter. So here we can see the TLR7. TLR7 recognizes single-stranded RNA which is present in virus, and TLR3 recognizes double-stranded RNA which is also present in virus. So they activate this TLR signaling. This is the in case of endosomal. This is here I am showing the plasma membrane one, which can directly sense the pathogens, but in endosomal one it will only sense the endocytosed pathogens. So here you can see this TLR7 is binding to single-stranded RNA and is then recruiting mediated, which again is in the similar pathway like recruiting IRAC4, IRAC1, then phosphorylation of IRAC1, which is recruiting this TRAC6 and TAC1 complex, which is then phosphorylating this. All these are kinases. This TAC1, IRAC1, IRAC4. So this is again recruiting, and this is again phosphorylating this complex Nemo IKK alpha IKK beta, which is then leading to the phosphorylation of I kappa B, which is getting degraded. Then NF kappa B is entering the nucleus and like it activates the transcription of different genes like interferon genes or ISGs, whatever. But in case of TLR3, you can see that it is uh, interacting with the adapter, TRIF adapter, and not mediated. So here also it is binding to TRIF, and it is activating the signaling pathway through a different pathway. It is not activating the NF kappa B1, but it is activating the IRF3, IRF7 uh, pathway in which first it is binding to the double-stranded RNA of the virus, this TLR3, which then signals through TRIF, this adapter, then TRIF activates and recruits TRAF3. Here you can see TRAF3, which activates the I kappa complex. That phosphorate and I activate both IRF3 as well as IRF7. How after that, IRF3 as well as IRF7 goes to the nucleus and it induces the transcription. These are transcription factors. It induces the transcription of several interferon genes like interferon alpha, beta. Here also again you can see these all signaling pathways are interrelated among each other. So here TRAF3, here one arrow is showing. So the TRAF3 can also like activate the TRAF6 and it can also lead to the NF kappa B activation. So it is not that whenever this signaling is occurring, this signaling is getting stopped. So there everything is interconnected. Here TRIP can also induce the MAP kinase pathway. TAC1 complex also can induce the MAP kinase pathway. So all these are interconnected and all these are uh, recruiting or activating the different molecules like interferons or any kind of ISGs. So there are two types of signaling. One is plasma membrane signaling, another is endosomal TLR signaling. And there are two adapters through which signaling occurs, mediated and TRIP. And this leads to the synthesis of different kinds of cytokines, chemokines, and antimicrobial components. So then this is about all about TLRs or the toll-like receptors. After this, we come to CLRs or C-type lectin receptors. These C-type lectin receptors bind carbohydrates on the surface of this extracellular pathogen. So they bind mostly fungal and bacterial cell wall components, mainly sugars and polysaccharides. And binding of these PAMs triggered a variety of distinct signaling pathways that activate the transcription factors that induce the expression of inflammatory cytokines. So here we can see two different types of receptors. Dectin-1, this is a type of C-type lectin receptor. Dectin-1, they this is a fungal cell wall and there are molecules which are present. They binds to these molecules which is a glucan and they dimerize and you can see there are motifs as in TLR there were TIR domain, here there is ITAM domain. This there are tyrosine present in this domain, that is immunoreceptor tyrosine based activation motif, the full form of ITAM, and this tyrosine gets phosphorylated by a tyrosine uh, kinase whenever there is activation of these receptors by any fungal component, and then it activates different signaling pathways like it can activate IRF5, can activate NF kappa B pathway, it can activate MAP kinase pathway, it can activate calmodulin calcineurin pathway. So you can see how many pathways it is activating by just binding to a single fungal component. Like a plethora of uh, uh, molecules are being produced, like all these transcription factors are going into the cell and transcribing, helping in the transcription of these cytokines, chemokines, or antimicrobials. So this also similar to the toll-like receptors can activate different types of molecules and lead to the activation of cytokines, chemokines, and antimicrobial compounds. After C-type lectin receptors, there are receptors known as node-like receptors. They are NLRs or node-like receptor. They bind pathogens from the cytosol. They bind PAMs from cytosolic pathogens. So they are also having different kind of protein domains like TLRs. They are also having leucine-rich lipid domain, and they have uh, a nucleotide binding domain NBD domain, which is present, and this leucine-like receptor, you can see these are the domains, LRR domains. This is the nucleotide binding domain. So this will bind to this LRR will bind to the ligand. So there are different types of NLRs which are present. Here you can see NIP, NOD1, NLRC4, NOD2, NLRC3, NLRC5. Different types are present and all are having different functions, different domains. So here you can see three bead domains is there, which is absent here. While here you can see two card domain is there, which is absent in this. So there are three main classes of node-like receptor which are distinguished by the N-terminal domain, like NLRC receptors, which are having cards domains. NLRP have pyridine domains, this PYD domains, and NLRB receptor has this bead domain, so baculovirus inhibitor repeat domain. So all these are having different kinds of domains, but the function is the same. There also it is through the homotypic domain interaction, like in case of TLR or in case of CLR, there is formation of homodimer or heterodimer. These not like receptors also can uh, interact with each other and form homodimers and can activate the downstream signaling. Mm, Here we can see the signaling by the node-like receptors. Like here you can see a dimer, node 1 dimer, node 1. Here you can see NLRC1 or node 1, which is having a card domain, NBD domain, and the LRR domain. So there is binding of this node 1 dimer with a bacterial component, which is a diaminopymalic acid, which is a fragment of cell peptidoglycan present in a cytosolic or endocytous bacteria. So here it is binding to some DAP component of a bacteria. There is dimerization. There is recruitment of some molecules DIP2, which then binds to TAC1 complex, similar TAC1 complex which we studied in TLR, which then activate the MAP kinase pathway or the NF-kappa B activation pathway. In another way, it can also, in dendritic cells especially, can bind also activate TRAF3, which lead to the phosphorylation. And as I told, in case of endosomal TLR, IRF3 and IRF7, and all these transcription factors move into the nucleus to regulate the transcription of these different types of cytokines, chemokines, ISGs, antimicrobial peptides. So all these are having the difference is that their structure is different, their binding affinity to the ligand is different, their but the downstream signaling process is almost similar and all are activating the almost the similar kinds of pathway. So these not like receptors contain many domains as I told. So when there is a domain and domain interaction, they allow them to assemble with other proteins to form large complexes. So these molecules will form large complexes which are known as inflammasomes. Why inflammasome? Because they are largely involved in inflammation. Uh, and Here we can see NLRP, NLRC. This NLRP especially can form large complexes and are termed as inflammasomes. So, following the binding of the PAMs or the other cellular proteins, they activate some proteases. Proteases means enzymes which cleave the proteins, which are known as caspases, which convert the inactive large precursor form of cytokines, which are known as procytokines, to active cytokines IL-1 beta and IL-18 into smaller mature forms. And It can also induce the death of activated macrophages through pyroptosis, allowing the release of the mature IL-1 beta and IL-18. So, because of the very potent inflammatory effect of the secreted IL-1 beta, these large complexes of enolars with caspases and other proteins are referred to as inflammasome. As I already told, like this, they are known as inflammasome because they, what happens when these node-like receptors will sense these spams or dams or whatever other proteins, and they will recruit some caspases, which are nothing but proteases. These proteases will cleave certain immature form of cytokines of IL-1 beta, IL-18 into a mature form, that can be secreted. And uh, whenever these kinds of like IL-1 beta, IL-18 are secreted, there is a potent inflammatory effect which is occurring inside the cells. So these large complexes of NLR and caspases as well as other proteins are referred to as inflammasome because of the inflammatory nature of this IL-1 beta and IL-18. So here you can see the structure. Caspase, this is the NBD, this is the ALR, NBD, PYD, this is the NLRP3 as I already told. NLRP family is the most important uh, NLR inflammasome. Here NLRP1, P2, P9, these are the NLRP family, and this is having this NBD or nucleotide binding domain, this pyridine domain. This is the NLRP3 node-like receptor, NLR. Apart from that, it is interacting with the caspase as I told already. Caspase is having card domain. This is a ASC, or AC is like another protein which actually recruits this to like helping binding of this protein, NLRP3 and caspase 1. So this is having a pyridine domain which is interacting with this PYD domain of NLRP3 and a card domain which is interacting with the card domain of caspase. So it is acting as a bridge between this NLRP3 and caspase 1, and it is forming a large complex. As you here you can see, these are the so many NLRP3 molecules interacting with so many caspases here, and it is a very much large complex which is forming, and this whole complex is known as inflammasome. So it is due to the aggregation and homotypic domain interaction between three component proteins, NLRP3, ASC, and caspase-1. And activators can be divided into two categories. Sterile activators include non-microbial self and environment-derived molecules. Pathogen-associated activators include PAMs derived from bacteria and protozoa. So these can be activated. This inflammasome uh, can be activated by different kinds of molecules. Like one can be the obviously the PAMs, which are derived from the pathogens from bacteria, virus, fungi, protozoa. Another is between like a non-microbial self and environment derived molecules. So there is recruitment of these molecules whenever there is a need to like remove this the pathogen, to remove the pathogens from the body, like excessive amount of pathogen is entering, then there is a need for this whole complex to go and clear these pathogens. And so here you can see the different pathogen activators include the pore forming toxins, flagellin, peptidoglycan, RNA, DNA, then virus derived means influenza M2 protein, RNA, fungus derived beta glucans, mannan, zymosan, hyphae, protozoa means hemozoin. All these can induce this inflammasome. Then other sterile activators include ATP, cholesterol crystal, urate crystal, glucose, amyloid beta, hyaluronan. These are the from the self derived, from our self only it is getting induced, these molecules. Then there are environmental derived like UV, skin irritants, silica, asbestos, alum. So whenever there is presence of these ligands, either from the pathogen, from the environment, or from ourselves only, there is a recruitment and formation of this inflammasome, which causes like this inflammation by mainly this IL-1 beta as well as IL-18. So here is the signaling of the inflammasome. Like there are two types of signals present here. We can see signal 1, signal 2. Signal 1 includes binding of a PAM to a PRR. Example is like TLR, which activates the NF-kappa B and the MAPK signaling pathways that induce the transcription and synthesis of large cytokines like pro-IL beta and pro-IL-18, and of the NLR-NLR-P3. This constitutes signal 1 for the generation of mature IL-1 beta and IL-18, as well as from the signal to various different sterile and pathogen-derived activators lead to the binding of some protein kinase NEK7 to NLRP3, triggering its assembly with ASC and procaspase 1 into NLRP3 inflammasome. Then procaspase 1 is cleaved into active caspase 1, which ultimately cleaves this IL-1 beta into mature form and IL-18 into mature form. And um, also it can induce gasdermin D, which can induce cells to undergo pyroptosis. So these are the different types of signals which can activate this NLRP3. So whenever there is a binding of PAM, it can induce NLRP3 form, NLRP3 inflammasome formation. When after binding of the PAM, there is PRR signaling pathway, which can induce NF-kappa B or MAP kinase pathway, and it can lead to the synthesis of certain cytokines, chemokines, or antimicrobial peptides. And out of that, it is inducing pro-IL-1 beta and IL-18, pro-IL-18. So pro enzymes need to be cleaved into their, these are the precursor forms, so it need to be cleaved into mature forms. So here, as you can see, NLRP3 inflammasome it is inducing. It will secrete the, here is the caspase, but caspase also is in the pro, like all the enzymes which are present in our system is uh, present as a pro or precursor form. So this precursor also needs to be cleaved, and when it is getting cleaved, it is becoming active and it is going and binding to this pro IL-1 beta and IL-18, and it is forming this mature cytokines. Mature IL-1 beta and IL-18. So apart from that, caspase 1 can also generate a fragment of gasdermin, which is another molecule that can induce cells to undergo pyroptosis. Pyroptosis is another like uh, type of apoptosis in which it is an inflammatory form type of cell death, which is triggered by microbial infection or host factor. Like it is a type of apoptosis only, which is caused by inflammation. Like whenever there is an inflammation, it will, the cells will try to degrade or try to die, and this gasdermin by active caspase 1 is promoting this pyroptosis. So this is the signaling through which this NLRP3 can work, and there is heavy inflammation for which the term inflammasome came. I hope no one is having any question, or if any question is there, you can ask me. You can unmute yourself, or you can ask me in the uh, comment section, the message section. So then comes, we came to know about the TLRs or the toll-like receptor, the RLR or the Rigai-like receptors, and the nod-like receptors, the C-lectin type receptors. Now we came to, are coming to know about uh, ALR or the AM2-like receptor. They are AM2-like receptors are also cytosolic receptor that can bind DNA molecule from both bacteria as well as viruses. They contain an amino terminal pyridine domain. They also contain two or more copies of the oligonucleotide binding domain or the heme domain, which is hematopoietic expression interferon inducibility nuclear localization domain at the C terminus, which acts as the DNA binding unit of the receptor. Thus, ALRs are sometimes referred to as the pi heme family. So, as you saw already, different types of receptors are having different domains. This is also containing one pyridine domain, heme domain, and which is at interacting with the DNA binding of the receptor, and they are often termed as a pi heme family because of the name pyridine as well as heme. So pi heme, it came from that word. And in case of both NLR and ALR, the pyridine domain serves as the effector domain, transmitting downstream signal to the cellular machinery. The prototype ALR M2 binds double-stranded DNA from cytosolic bacteria and viruses via its heme domain. The nucleotide binding domain, as I already told, is a heme domain. Here you can see the pyridine domain. In case of no NLR, is the pyridine domain. So this is binding to pyridine domain is binding to the different effector molecules. Here also similar thing is happening. So by this ALR bind to the same strand of double-stranded DNA, allowing their pyridine domain to associate, driving assembly into long filament that together with the protein ASC and procaspase-1 from inflammasomes and generate mature IL-1 beta and IL-18. Not only NLR but ALR can also form inflammasomes. By a similar way, these ALRs bind to this ASC and procaspase-1 form inflammasome, and they also generate mature IL-1 beta as well as IL-18. So this is one type of ALR like M2. M2 is one type of inflammasome which or the ALR which can form in inflammasome. Another ALR is IFI16. It is also known to bind viral DNA in either the cytosol or the nucleus. Its signaling pathways are still being characterized but may include inflammasome activation of inflammatory cytokine as well as other pathways activating IRFs and interferon production. So there are two types of uh, things like M2 and IFI16. These are the two different types of ALRs which are having different domains, and it also activates the it can also form inflammasome and generate this mature IL-1 beta and IL-18. So mainly these are activated by binding long DS or double-stranded DNA from cytosolic bacteria and viruses. On binding, they form inflammasomes that promote inflammation, and some can also induce interferon production. So as you can see, the downstream signaling is almost similar. This all these receptors are binding to the uh, ligands. Then there is other molecules that are coming up, and there is downstream signaling which is getting activated, and they can induce the formation of either inflammasome or they can also produce interferon. So after this, there is another important type of receptor which is Ligui-like receptor which is present. So Ligui-like receptors are nothing but RNA helicases that function as cytosolic PRRs that recognize viral double-stranded RNAs. So they act as helicases and are present in the cytoplasm, and they can recognize viral double-stranded RNA. While here we can see ALRs can recognize viral double-stranded DNA. But mainly, like recently, some studies came like in case of IFI16, it can recognize both DNA as well as RNA. So more and more studies are coming up on these receptors. So after PAMP binding, RLS triggers signaling pathways that activate IRFs and NF-kappa B, inducing the expression of interferon and cytokine. So RIGI binds double-stranded RNA via terminal 5' triphosphate, whereas MDA5 binds the body of double-stranded RNA molecule. So there are two types RIGI-like receptors. One is RIGI and another is MDA5. RIGI can bind to the there are 5' triphosphate present in the virus, so it can recognize those 5' triphosphate, whereas MDA5 can recognize the whole dsRNA or the double-stranded RNA molecules. So what happens? On viral binding, RNA binding, this undergo conformational change. So they are having helicase domain. So this helicase domain can bind to the RNA. So here, instead of forming a homodimer or a heterodimer, four RLRs form a tetramer, and then larger assemblies form a filament on that RNA. And they recruit multiple copies of adapter molecule. One is the like one adapter molecule is the MAPS or the mitochondrial antiviral signaling via association of their card domain. So these adapter molecule, like in case of MyD, TIT, and TRIF, in case of TLR, here it is MAVS or IPS1. So these MAVS protein again recruit other proteins like TRAPS, leading to the activation of NEMO, I kappa A, I kappa B, TBK1, I kappa KK, epsilon. So similarly, there also we saw like via chip, via my DTA, it activated the signaling. Here via MAPS, it is activating the downstream signaling. So as in other PRO signaling pathways, these also protein kinases activate NF-kappa B, IRF3, IRF7 molecule or transcription factors, and thus inducing the expression of potent antiviral proteins IFN alpha, IFN beta, as well as other cytokines. So here we can see the signaling um, process by ADLR or RIGI. This is recognizing the triphosphate, 5' triphosphate present in the virus. There are different domains here you can see. This is the CTD or the C-terminal domain. This is the helicase domain. This is the card domain present. So here the guy, as I told, it is forming a tetramer and forming a filament. And here this from the virus, this RNA is there inside the helicase domain. This card domain is helping to recruit the adapter protein. So in the the C-terminal domain of the RIGI interacts with the duplex RNA helicases, which is having a nucleoside triphosphate. Then it thus allows a conformational change, allowing the helicase domain to bind, HEL helicase domain to bind. Then there is association of multiple RIGI like receptors with the RNA forming a filament. This is a filament which is forming. Then this card domains bind to the card domain of the SMAS protein or the IPS1 or the mitochondrial antiviral signaling protein, recruiting different types of molecules like TRAPS, TBK1, I kappa K, leading to the activation of three different pathways similar to the TLRs like IRF3 pathway, IRF7 pathway, and NF kappa B pathway, and which is then entering into the nucleus and inducing the transcription of these interferons like cytokines, chemokines, and antimicrobial compounds. So all this, as I already told, are having all the pathways are converging at the same place. Only the different receptors are recognizing different ligands and they are having different structures. So these are the different types of receptors which I told. Apart from this, there are certain molecules which are known as C gas and sting. So they are cytosolic PRRs, pathogen recognition receptors, cytosolic PRRs, C gas, cyclic GMP AMP synthase. They recognize cytosolic DNA, usually of viral or bacterial origin. So, C gas is basically nucleotidal transferase. That is, after it is binding to double-stranded DNA, it is activated to synthesize a molecule CGAMP from GTP and ATP. It is getting like modified into CGAMP molecule. This CGAM then is serving as a secondary messenger binding the endoplasmic reticulum membrane associated protein STING, that is stimulator of interferon genes. It was previously identified that STING has the ability to bind to cyclic dinucleotide products CDGMP and CDAMP released from intercellular bacteria. Hence, STING is also a cytosolic PRI. So binding of cytosolic nucleotides alters the conformation of STING dimers, which then goes to the Golgi complex and recruit and activate TBK1. As similar to PRR signaling, CBK1 phosphorylate and activate the IRF3 NF-kappa B, leading to the synthesis of type 1 interferon and cytokines. So, C-gas and STING are also some cytosolic receptor which are present, and C-gas, after recognizing the double-stranded DNA, gets modified to C-GAM. Then C-GAM uh, stimulate the STING, then STING then activates this downstream signaling molecule where it activates TBK1 and activates the IRF3 and NF-kappa B signaling. Then similarly lead to the synthesis of type 1 interferon and cytokines. So here you can see the signaling. Here you can see C-gas in cytosol. It binds to the double-stranded DNA, either viral or bacterial origin. Then it gets activated to form C-GAMP. Here you can see there is an exchange of AMP to GMP and it is forming C-GAMP. Then C-GAMP then binds to this ER associated protein STING, stimulator of interferon genes. This STING is then binding to CD-GAMP, and other cyclic dinucleotide products CDGMP and CDAMP released from intercellular bacteria. Here, CDGMP, CDAMP released from intercellular bacteria. Then binding of these dinucleotides alter the conformation of STING dimers, where it can which recruit this TBK1 and can activate like TBK1. Then TBK1 phosphorylase IRF3 or this I kappa B, which is then degraded, and this IRF3 goes into the nucleus, and this released NF kappa B also goes into the nucleus to leading to the synthesis of type 1 interferons as well as cytokines. So all again it is converging at the same place, like it is activating the IRF3 and the NF kappa B signaling pathway. So anyone is having any question? Okay, so Induced innate immunity. So here comes the end of all the PRRs or pathogen recognition receptors and how they mediate the downstream signaling to clear off the pathogens. Now we will come to about come to know about the how the what is the effect of this innate immune system on other things. Like this is the overall view. Like this microbial invader can enter through the lesion in our epithelial barrier and which can expose the invaders to attack by various effector molecules and cells. There are different expression of different types of molecules, antimicrobial pro-inflammatory molecules, which is induced by extracellular pathogens recognized by either endosomal or plasma membrane PRRs, and also intracellular pathogens, which are recognized by cytosolic PRRs. So, extracellular pathogens with surface components recognized directly by PRRs or by soluble opsonins like C-reactive protein, mannose binding lectin, complement component surfactant proteins, all are cleared by phagocytosis. So these are the different types of molecules. We will come to know about complements in the next class. And PRLs also activate the production of pro-inflammatory cytokines, chemokines, increase the vascular permeability, allowing influx of fluids and cells like neutrophils or macrophages to cross the blood vessels and into the site of infection and further activate the innate immune cells. Pathogen recognition by PRLs also initiate adaptive immune responses. For example, dendritic cell bind microbes via receptors and are activated to mature. They also internalize and degrade microbes. They also migrate through lymphatic vessels to the nearby lymph nodes. Add their present antigenic derived peptides and the MHC to T cells. Antigen activated T cells then initiate this immune response against the pathogen. So these are the overall view by which there is the effector mechanism by the induced innate immune system. So here you can see first pathogen is coming, extracellular pathogen, either it is phagocytosed or it is opsonized by different types of molecules, complements, etc. Or there are antimicrobial proteins and peptides like CRP, complement MBL, which induce the lysis of the cells and which can degrade the membrane of the pathogen and can kill the bacteria or the pathogen. And otherwise, there can be molecular patterns present on these pathogens, which is recognized by these cells and receptors, which is then inducing the formation of this inflammatory response by inducing the secretion of the cytokines, chemokines, and also there are other IFNs and other antimicrobials inhibit and kill intracellular pathogens, and virus infected cells can be killed directly by natural killer cell. And also these chemokine cytokines which are secreted by these already infected cells can initiate the adaptive response. Here we can see dendritic cell PRRs recognize the PAMs, activating phagocytosis. They migrate to lymph nodes carrying intact or degraded pathogens. The antigen fragments bound to cell surface MHC are recognized by the T cells. With co-stimulation, activated T cells initiate adaptive response. So here is the overall mechanism by which our activated innate immune system can actually help in degradation of the pathogens. So as I already told, like there are different kinds of proteins like antimicrobial proteins or peptides, cytokines and chemokines which are being secreted by the signaling pathway. by all the different types of PRRs like the NLR, TLR, RLR, CLR, all these signal, all these receptors are actually activating the downstream signaling process, which are synthesizing common products like antimicrobial components like defensins, cathelicidin, which are produced by this epithelial like respiratory, intestinal, reproductive, keratinocyte, kidney, neutrophil, NK cells. They act on the pathogens, monocytes, immature dendritic cells, T cells, mast cells, and they have can inhibit or kill. They can be chemoattractant. They can activate cytokine production. They can activate degranulation. Then there are obviously interferons, interferon alpha and beta, which are the type 1 interferon, which are produced by the NK cells, macrophages, dendritic cell, virus infected cells. They act on these virus infected cells, NK cells, macrophages, T cells, and can inhibit virus replication or activate the NK cells, then regulate the activity of these different kinds of cells. Then different kinds of cytokines are released like IL-1, IL-6, TNF alpha, GM-CSF, IL-12, IL-18, IL-10. So mainly IL-1 is secreted by monocytes, macrophage, dendritic cell, keratinocyte, epithelial cell, vascular endothelial cells, and they act on lymphocytes, bone marrow, vascular endothelium, liver, hypothalamus. They enhance the activity of lymphocytes, promotes neutrophil production, activate increases vascular permeability, induces acute phase response, can also cause fever. Then IL-6 are also similarly secreted by these kind of cells. They also more or less their active like function is also similar to this IL-1. Then TNF alpha also is more or less similar. They are secreted by macrophage, vascular endothelium, liver, hypothalamus, tumor. They can activate the macrophages, increase the vascular permeability, fluid loss, local blood clotting. They can induce acute phase response. They can promote the fever. They can also be cytotoxic to many kind of tumors. Then GM-CSF is a granulocyte monocyte factor, which is produced by macrophages of vascular endothelial cells. They can act on bone marrow, they can stimulate hematopoiesis, uh, and they lead to the formation of the myeloid cells mainly. Then there is IL-12, IL-18, which are produced by monocytes, macrophages, dendritic cells. They act on NAP, CD4, and CD8 T cells as well as NK cells. They induce Th1 phenotype, IL gamma production, and activate these NK cells. Then there is IL-10. IL-10 is basically the anti-inflammatory molecule compared to all the other cytokines. They are produced by macrophage, dendritic cell, mast cell, NK cell, T cell, B cell. They also act on macrophage and dendritic cell. They antagonize inflammatory response. That is, they are anti-inflammatory in nature. They produce IL-12 and Th1 cells. Then there are chemokines. Chemokines means which are actually uh, like cytokines. They are small molecules which actually are acting as a chemoattractant to like bring the different kinds of cells to the infection site. So they are also produced by macrophage, dendritic cells, vascular endothelial cells, etc. They act on neutrophil, basophil, immature dendritic cells, and T cells. They help the cells, they chemoattract the cells to the infection site. So these are different kinds of chemokines, cytokines, antimicrobial peptides, which are produced by PRR signaling and helps in innate immune response. So that's all for today. So if anyone is having any question, they can ask me.
Good evening, ma'am.
Yes, good evening.
Ma'am, actually, I wanted to ask. We have studied RLR signaling and rig signaling pathway, ma'am.
Yeah.
So I wanted to ask how the questions can be asked in the exam. like we have to memorize the whole signaling pathway, or is there any other way to memorize it? Because uh, I find it quite challenging. For
Okay.
me, it's challenging, yes, ma'am. So, how the questions being asked in the exam, or in what way we have to like write or explain the when questions like these will come.
It will be basically MCQ questions only, and you don't need to memorize the whole signaling pathway. Just remember which are the adapters present, or you can remember like what is sensed by what receptors. Like different kinds of TLS senses different kinds of ligands, different kinds of RLR signal, like as I told, Rigai senses something different, MDFI. What are the ligands, different ligands which are sensed by these receptors? You can just just have a look. Like you don't have to
Yes,
memorize
ma'am.
also. Just you
Hmm.
while going to the slide, you can just remember like this is interacting with this ligand, this is interacting, and whole signaling pathway. If you see also all these like receptors are converging at the same point. Either they are activating the MAP kinase pathway or the IRF3 pathway or the NF-kappa B pathway. So just if you see as a whole, then you can see that all are converging at the same time. Just what is different between them is just the domains which they are having, and you don't need to remember like what are the domains present or in each of the receptors, but at least try to remember what are the different types of ligands which are which are recognized by these different kinds of PRRs.
Okay, ma'am. And one more, ma'am. We are facing, ma'am. I have some issues in the assignments. Like, I find some answer to the some questions being wrong, और मैम मैं हो सकता है मैं गलत भी हो सकती हूँ, because maybe I have skipped some part. So
Huh.
can I don't remember at the moment what are those questions, but can I ask you the in the next lecture those
Yeah, you
questions?
can
And
ask or you can post also in the portal. There you can ask any question. The portal also you can write what is your query, then we can answer it. Or in the next class also you can ask.
yes, ma'am. Actually, the answers seem wrong to me, but I don't know. Maybe I might be wrong, but that's why. I don't remember. I have like one or two issues in the questions in the assignments. So uh, I will ask you in the next lecture. And ma'am, one more thing. Can we get these presentations from which you are teaching us?
Yeah, I will be uploading it in the Google Drive itself. So you can get it. I think they will give the Google Drive link and there you will have all the like week 1, week 2 lectures.
Oh, okay, ma'am. Because, ma'am, yesterday I was going through the presentations which are on portal, and those were quite different from what you were teaching us.
Yeah, this is kind of what Sir is teaching. Sir has covered the whole kind of immunology thing. But what I'm teaching is like some more portion to it. Like
Yes, ma'am.
so you just you don't need to memorize my whatever I am teaching, but you just memorize what Sir is teaching. I am just adding some extra things which you might find interesting to know or something because the whole
Actually,
immunology means huh.
yes, ma'am. Actually, yours is a bit easy to understand because you have like clearly explained the everything, like the terms and the signaling pathway in a very
Yeah,
brief
if
way.
you find, if you find like that you cannot understand, you can follow my slides also. You can see my slides, and if you can try to understand. That is not problem. But I am just teaching whatever extra is there or something, just to make it like more interesting.
Okay, 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 ma'am.
And if
Okay,
you're
ma'am.
having any doubt, you can either post in that portal because there will be options, or you can ask me in the next class.
Okay, ma'am. I'll ask you in the next lecture.
Yeah, yeah, sure. Which week you remember? Which week you are having any problem?
Ma'am, actually, till now we have done like 5 assignments, and I think there are 2 or 3 questions I clearly can't remember. And while I was going through the solutions, I was like, I thought this was the answer, but it turned out to be wrong. So I just wanted to know why this
Okay.
can be the answer. Yes, ma'am. And why
Or you
only the
can,
option this is the option. Yes, ma'am.
you can mail me also if you remember. I am writing my mail in the.
Yes, ma'am. I have your mail actually. I have your but
There
what
also you
but
can. Huh.
ma'am, but right, ma'am, just you can share your email once again. I have it. I
Huh.
remember, but just ma'am, please share for once.
I am writing in the. You can writing in the message box. You can just mail me about the questions. Then next class I can uh like I answer the queries.
Okay, ma'am. Okay, ma'am. Thank you, ma'am.
Thank you.
Thank you, ma'am.